Wound Healing, the Basics on the Cellular and Biochemical Levels, Hemostasis, Part 1. For our discussion, a wound is defined as a break in the skin's integrity that causes the formation of a pathway for the outside environment to enter the internal environment of the body. The reestablishing of structural integrity of the skin is part of the healing process. Now, when a scalpel blade makes a cut, the integrity and the continuity of the skin structure, its epidermis and the dermis and the blood vessels in the dermis, is disrupted. Healing begins in each of these areas where the disruption of the skin structure has occurred. Let's say at the time of surgery, when a scalpel blade lacerates the skin, the outer layer of the epidermis is cut first. There is no bleeding because there are no blood vessels in the epidermis. This is how the surgeon knows how deep an incision is. But eventually the laceration cuts below the epidermis and into the dermis, where bleeding does occur because the dermis of the skin is where blood vessels do exist. So what is happening as the body heals from this laceration? The first step is to control the bleeding. Let's begin with some background information about the anatomy of blood vessels. Remember, the blood vessels have a structure that can be found in any textbook of histology. Histology is the microscopic study of tissues. The structure of blood vessels can be thought of as in layers. The innermost layer of any blood vessel, the artery side of the capillary itself, or the venous side of the capillary, is the endothelial layer, which is named after the cell that makes up the layer, an endothelial cell. This is a very thin layer. In fact, it is one cell layer thick, and the cell itself is very thin. These cells form a continuous layer along the inside of the blood vessels. These cells adhere to each other so that only the endothelial cell surface contacts the blood within the lumen of the blood vessels. These endothelial cells are supported by collagen below the endothelium called the subendothelial collagen. This is also a very thin layer. The outer structures of these blood vessels depend on the type of blood vessel it is. An artery, or arterial, which is a small artery, have a well-organized muscular layer of smooth muscles. Capillaries do not, and veins or venules, which are small veins, have a muscular layer but is less developed than the artery side. Looking through a microscope, the artery looks very round because of the thick and developed smooth muscle layer. The vein is not round at all because of the poorly developed smooth muscle layer. Smooth muscles are called involuntary muscles because one cannot cause these muscles to contract consciously, like, let's say, the pectoralis major muscles, which are voluntary muscles. In the oxygen delivery lessons, we spoke of red blood cells, RBCs, because these cells carried oxygen within the blood. But there are other cells in the blood, and there are also proteins in the serum of the blood. For any discussion of blood clotting, an important cell is the megakaryocyte. This is a very large cell. In fact, it is 10 to 15 times the size of a red blood cell, and it is the source of blood platelets, and platelets are key to the formation of a blood clot. Platelets are tiny cells located in the blood that are actually small pieces of a megakaryocyte. These tiny cells have a cell membrane, and the platelets contain granules within their cytoplasm. Proteins in the serum of the blood are also very important for clot formation. With this background, let's explain how the clot forms. Remember, it is a dynamic process, and it begins with the disruption of the integrity of the blood vessel wall. So what is happening to the anatomy of the blood vessels when the blood vessel is disrupted? Quite simply, the smooth muscle in the wall of the blood vessels contract, making the size of the lumen smaller, and the endothelial lining is disrupted, which exposes the subendothelial collagen to the blood within the blood vessels. This is where it all seems to begin. Sequence of events. The blood vessels are injured because of the formation of a wound, which cuts the epidermis, the dermis of the skin, and the blood vessels within the dermis. Let's say the structures are cut here, and let's enlarge the diagram of a blood vessel. Vasoconstriction is the first response of a cut blood vessel. That is, the blood vessels will constrict due to the lack of continuity of the structure of the blood vessel. This has been described as a reflex of muscles contracting from the outer layer of the blood vessels. This decreases blood loss by making the cross-sectional area of the blood vessel smaller. The subendothelial collagen that is exposed to the contents of the blood initiate a sequence of events that becomes very confusing rapidly as the process initiates a whole series of cellular and biochemical events. Each event is linked to another event. The next event is the formation of a platelet plug. The formation of a platelet plug is a key initial event. Remember, platelets are tiny cells located in the blood that are actually small pieces of a larger cell called a megakaryocyte. These tiny cells have a cell membrane and contain granules within the cytoplasm. When the subendothelial collagen is exposed to the blood, these platelets adhere to the subendothelial collagen, and other platelets attach to the platelets already contacting the subendothelial collagen, thus forming a plug. This is called the platelet plug. This platelet plug helps seal the blood vessel from further blood loss. So when a blood vessel is cut, the blood vessel's muscular layer constricts as a reflex. Platelets adhere to the collagen below the endothelium, forming a platelet plug to seal the leaking blood vessel. Up to this point, the process is reversible and is called primary hemostasis. To have a platelet plug that is reversible needs something more permanent, so a sequence of other reactions take place on the cellular and the biochemical level. There is a chemical called adenosine diphosphate, ADP, which is released from damaged tissue and from platelets. Another substance called platelet factor 4, which is found in the serum and in the presence of calcium and magnesium, cause platelets to release its granules. This requires another substance called fibrinogen for this to occur. Fibrinogen will be discussed shortly. When all this occurs, the platelets become compact, forming an amorphous plug. This is considered not reversible. With release of these substances and another substance called platelet factor 3, a series of steps begin, which is called the clotting cascade. More on this in Hemostasis Part 2.